Hi there, everyone. My name is Trini Taccad, who is a senior undergraduate microbial biotechnology student who is currently working in Dr. Brad Hillman's laboratory together with Alanna Cohen and Limei. We are interested in these viruses because they may affect biology of the fungus and they may have potential for biological control of the disease. Dr. Hillman initiated the project last year with Limei in the lab since by November. In this case, today I will be presenting our poster presentation titled Viruses and Fungi that Cause Dollar Spot Disease of Turf Grasses. In this slide, I will provide you the general overview of today's presentation. First, I would like to start with some introduction, providing you some general background of the topic and a few previous studies that were involved to start this project. After that, I will go over the methods that we did towards the goals of the project, followed by data results, and lastly, I'll go over the discussion and conclusions regarding about the experiment progress towards our objective and what are the current limitations that we're trying to assess prior to moving to our next step of the project. And with that, let's go over to some introductions. Dollar spot, as we know, is a highly destructive and globally distributed disease to a broad range of C3 and C4 turf grasses, cool and warm season turf grasses. This became a focus of Rutgers turfgrass breeding and disease management efforts. This disease is caused by several related ascomycete fungi. Earlier studies by Joan Crouch and Bruce Clark led a group, and the taxonomy of dollar spot fungi was re-examined. With their research, many of the disease-causing isolates were reclassified out of the genus Sclerotinia and into the newly established genus Sclerotia. Past efforts by Greg Boland and colleagues at the University of Guelph identified viruses that reduced virulence of the dollar spot fungi. The objective, or the general objective, is that we set out to take advantage of the collection of organized and well-characterized isolates of Sclerotia available through these previous studies to re-examine the role of viruses in fungal virulence. Here is an example of a severe dollar spot in Kentucky bluegrass turf plot in the left and the leaves on the right. We set out the first bioanalysis of double-stranded RNA, which is the signature of fungal viral infection. Then, we analyzed small RNA populations that may contain virus sequences by next-generation high-throughput sequencing of fungal isolates. Third part of the study is correlating the presence of the virus with effects on fungal growth and virulence. And as of today, we're at the second part of the study, which in this presentation, we will get to our methods of our constructed novel microbial discovery pipeline. As we finish the introductions, let's go over the methods. Again, we took the Sclerotia isolates from previous studies and performed a small RNA extraction and small RNA library preparation to get small RNA libraries. Then we sent the library for sequencing. From this point, we will proceed to our viral discovery pipeline using bioinformatics. We took the sequences and mapped them with the FIX174 spike-in as a reference genome using STAR and removed the Illumina adapters using CutAdapt to get phage-free reads. We then took those reads, performed a quality assessment using FastQC, and quality trimming with SYCL, which we also stated to remove 15 base pairs and below, which in this way, now we have high-quality paired and reads. We again use STAR mapping to map the reads with its host genome, and therefore, we have reads that are not part FIX174 and host. We perform a de novo assembly using Trinity with the leftover paired and reads to obtain assembled sequences. We then used transdecoder to translate the nucleotide sequences into amino acid sequences. The amino acid sequences were then blasted against the RefSeq viral protein database, and we identified sequences with viral hits. We pulled out the sequences that hit to ormanovirus, rhinovirus, and mitovirus, and aligned them together with the annotated viral sequences from GenBank. With that, we now have aligned sequences with viral hits. Finally, with those aligned sequences, we generated a phylogenetic tree using IQ tree. And with that, let's go over to the data results. As I said earlier, the first part of the project is by performing a bioanalysis of double-stranded RNA of the isolates to determine the presence of mycoviruses. This is the image of the double-stranded RNA extraction gel of some of the Claridia isolates taken by Limei, which in this case, there were visible dark to faded bands on the gel, indicating that the double-stranded RNA extraction protocol did extracted double-stranded RNA from the isolates, which tells us that there were mycoviruses present in several of the isolates. In this slide over here, this is the data I pulled out from one of the FASTQ quality assessments and sickle trimming of the reads. As you can see on the left side of the figure, that many of the reads with sizes ranging from 40 base pairs to 151 base pairs fall within the mid-yellow and bottom red areas of the graph. This indicates that they have acceptable to poor quality scores, and reads that fall within the green areas of the graph indicates excellent quality scores. After performing quality trimming with sickle, as you can see on the right side of the figure, that most of the reads fall within the green areas, indicating the reads had been trimmed to have a higher quality, and sizes 15 base pairs and below were discarded. Here's the table data summary I constructed to show you the star mapping output that I got when I mapped all the reads with their host genome as a reference in all of the isolates, and also the number of de novo assembled reads using Trinity, a de novo assembler, and by performing a BLASTP analysis, I counted the number of sequences identified to have a viral hit, specifically to Narnavirus, Ormeavirus, and Mitovirus. I also wanted to mention here that there were two species of Claridia that were analyzed in the pipeline, such as Claridia jacksonia and Claridia montidiana. But we can ignore the species difference, for we are looking for viruses that infect daughter spot disease-causing fungus, which all belong to the genus Claridia. This is the result of our multiple sequence alignment, which here we took at least two of the assembled sequences with viral hits and aligned them with similar viruses. Looking at the top figure, the alignment of sequences had shaded regions that shows the degree of similarity. The darker the shading, the more conserved it is. This shaded sequence is very well conserved, and it is identified as a protein superfamily, which in this case belonging to the myrovirus RDRP superfamilies. From the multiple sequence alignment, Alanna reconstructed a phylogenetic tree to determine not only what these new sequences are most similar to, but how are they related to other fungal viruses. This tree is a simplified representation of a maximum likelihood tree, reconstructed from a multiple sequence alignment of many of the full-length RNA-dependent RNA polymerase protein sequences from members of the mitovirus, ormeavirus, and Narnavirus, using Levivirus as an outgroup. We recovered a well-supported monophyletic clade for mitovirus. The literature supports two major clades of mitovirus, mitovirus A and mitovirus B. And while we recovered both of these clades, there are some unresolved relationship among the more basal relationship within mitovirus. We also recovered a monophyletic clade with ormeaviruses, sisters to the Narnaviruses. This relationship between the three genera are consistent with the literature on Narnaviridae. As far as our newly recovered sequences are concerned, they are distributed among the entire three. The vast majority of our sequences fell into ormeaviruses. We did recover a total of 17 sequences that belong to myrovirus, and those sequences are present in both major clades. In addition to the putative ormeavirus and mitovirus sequences, we also recovered 9 sequences that hit to negative-stranded RNA viruses, which in this slide, Alanna reconstructed a phylogenetic tree of negative-strand RNA viruses. Most fungal viruses contain either a positive single-stranded or double-stranded RNA genomes. However, in the last 10 years or so, negative single-stranded RNA viruses were identified in fungi, leading to a new family, Maimonaviridae. We used a similar approach, but expanded our sampling to include a wider range of viruses from related negative single-stranded RNA virus families. In this case, all of our sequences that hit to negative-stranded RNA viruses fell into a well-supported Maimonavirus clade, along with other negative-stranded RNA mycoviruses of Sclerotinia and Fusarium. I will now proceed to discuss the conclusions that we had led to in the progress of our project, also discuss about the future works that the lab plans on doing towards our objective. For conclusions, we have performed two different methods to identify viruses in several of the isolates, which was the first and second part of the study, such as the molecular work done by Limei to determine the presence of double-stranded RNA in some of the isolates, and using a bioinformatics approach with the constructed novel viral discovery pipeline. Because of this, we found possible viral sequences, and some of which were similar viruses known to infect related sclerotinia. For future work, that goes down to the third step of the study, which is to correlate the presence of these new identified viruses with the effects on fungal phenotype. To do that, we will extract these viruses and infect them to uninfected Chlorida isolates. But prior in doing that, we had to generate uninfected isolates by means of hypotyping and or single conidia isolation. And with that, and here is the reference slide that I used to provide support for this presentation. Thank you for listening, everyone, and have a good day.